Hi and welcome to my OCR A-Level Biology revision session with me, Christine. So today's lesson I want to look at limiting factors, and that's part of your photosynthesis module. So let's just have a look and remind ourselves about the light dependent reaction. So the light dependent reaction then is where these photon of lights are going to be absorbed by our photosynthetic pigments. Electrons become excited, they jump up their energy level, and they are able to move through the electron transport chain. That is going to pump hydrogens into the thylakoid space, and that's also going to result in reduced NADP being produced for the Calvin cycle. Now those accumulation of hydrogen ions are going to move through ATP synthase by chemiosmosis, and that is going to provide the necessary energy to form ATP. So ATP and reduced NADP are products of the light dependent reaction, and to replace the electrons that were donated from photosystem 2 to photosystem 1 we require photolysis to occur, the splitting of water, so that those electrons can be taken back into the photosystem 2. So therefore it's also producing oxygen as a waste product. So those products that were produced by the light dependent reaction, some of them are needed in the light independent reaction. So reminding ourselves that we have carbon fixation, RuBP and carbon dioxide are fixed into a 6-carbon molecule by the enzyme Rubisco. That will then lyse itself into GP, the glycerate 3-phosphate. The GP will then be converted into TP through a reduction process using those products from the light dependent reaction. One TP molecule can go off to become any of the other substances that are needed by the plant, and the rest need to be regenerated back into RuBP, and that requires ATP, which again is a product from the light dependent reaction. So if we then look at limiting factors, and what are limiting factors? Limiting factors are these factors that are going to reduce the rate of photosynthesis. So if they are in short supply, they are going to reduce that rate. They therefore are called a limiting factor. Now it's quite obvious, if we're looking at the light dependent reaction, we require light, those photons of light, to be absorbed by those photosynthetic pigment. So they are going to limit the rate of photosynthesis. They are going to limit the light dependent reaction. If there's no light or the light levels are too low, the light dependent reaction is going to cease or decrease, and that is going to result in less of those products for the light independent reaction. So whenever you look at a graph, before you get to the fact where it plateaus, whatever is at the bottom on the x-axis, that is your limiting factor. Well, let's look at what other factors might be limiting the rate of photosynthesis. So if I tell you that this first line in my graph is showing you that there's a low carbon dioxide concentration and low temperature. Now, carbon dioxide concentration, remember, carbon dioxide is necessary for the light independent reaction because carbon dioxide is fixed, that carbon is fixed into the 6-carbon molecule through the process of Rubisco. Now, Rubisco is an enzyme, so we're talking about a low temperature. So that low temperature is going to mean that there's less kinetic energy, but what's the limiting factor in this case is actually that carbon dioxide. If there's not enough carbon dioxide, there won't be enough carbons to be fixed to convert the RuBP into GP. So if we increase the temperature, if we increase the carbon dioxide concentration but keep the temperature low, then what we have done this time is we're no longer having that carbon dioxide is our limiting factor. What we have is that the photosynthesis is being limited by temperature. So light intensity then, if it's light intensity that's our limiting factor, that's going to be limiting the light dependent reaction. If we're talking about carbon dioxide concentration, we're going to be limiting the light independent reaction. if we're talking about temperature, temperature is involved in these enzyme controlled reactions. So when we affect the temperature, we have to take a step back and remind ourselves that if there is low temperature, there's going to be less kinetic energy, therefore less successful collisions, therefore a lower rate of reaction. We increase the temperature, we're going to have more kinetic energy, more successful collisions, a higher rate of reaction. Well, until those proteins become denatured. So whether we're talking about the proteins which are part of the light dependent reaction, which are necessary in the electron transport chain, or whether we're talking about the enzyme rubisco, and if we denature that, we affect that active site shape, then we need to be aware that if the temperatures are too high, the plant will not be able to photosynthesize effectively. But at no point in my graph do you see the fact that the graph is actually dropping down. The rate of photosynthesis is dropping down and it's not continuing to go on. And what could that be then? So if I increase the temperature, if I have higher concentrations of carbon dioxide and I have higher temperatures, what is going to be limiting that rate of photosynthesis? Because we've still got a higher rate of photosynthesis, but it's starting to plateau because it's starting to get to a level where it is being limited by something else. So let's just have a think about what that other factor might be. So let's think this one through then. We know that stomata are open so that carbon dioxide can diffuse into the leaves and so that it can be used in the light independent reaction. Well, as a consequence of stomata being open, water vapor will be lost. So the evaporation of water, which changes it from the liquid state to the gaseous state, the diffusion of that water vapor then out through the stomata is known as transpiration. So when there is a dry spell, when plants experience what's known as a water stress, it will actually prompt them to close their stomata. They need to reduce that water loss through transpiration. Well, if they're going to close that stomata, what they're now doing is they are limiting the carbon dioxide intake. Now, limiting that carbon dioxide intake is going to affect photosynthesis efficiency. So let's just remind ourselves about how the stomata are actually closed, because this is part of your module 2, differentiation of cells. So we know that we have guard cells which surround our stomata, and what we know is that those guard cells can be signaled to actively transport potassium ions out of their cells. Now, if they are actively transporting these potassium ions out of their cells, that's going to decrease the solute concentration and raise the water potential. That's going to mean that water will leave by osmosis. That will cause the cells to become flaccid, and because they have become flaccid, that will close the stomata because the water will leave by osmosis. And if water leaves by osmosis, the cell is no longer turgid, it's now flaccid, we close that stomata. Well, if we've closed that stomata, we are therefore affecting the carbon dioxide concentration. We know that carbon dioxide is needed for the light independent reaction, and rubisco will fix that carbon into the RUBP and we will create GP. Well, we know that oxygen is produced by the light dependent reaction, and we know that that oxygen is going to be needed for respiration. Well, what you need to understand is that oxygen is a competitive inhibitor, and because it's a competitive inhibitor of rubisco, when our carbon dioxide levels are low, remember those stomata have now been closed to save water. Also, if there's an increase in temperature, we've got more kinetic energy. More kinetic energy means that the competitive inhibitor is going to also have more kinetic energy and therefore could be competing for rubisco. So what happens is if oxygen binds to rubisco, it actually leads to the production of a phosphoglycolate. Now that is a toxic substance that needs to be removed. Therefore, energy from ATP is required to convert the phosphoglycolate into a useful metabolite, such as glycerate, which can then re-enter the Calvin cycle. Now, obviously, photorespiration is going to produce carbon dioxide as a product, but that carbon dioxide is not going to be enough to help with the low levels that are currently resulting in more photorespiration. So water is not called a limiting factor of respiration. However, it will have an effect on the rate of photosynthesis, because if there's not enough water, that's going to cause the stomata to close. Those stomata closing is going to affect that carbon dioxide concentration. So I hope you like this video, and if you do, then please do click on the like button and subscribe to my channel. Also, do check out my revision platform, www.aiqchat.com.